The enzyme Rubisco. It's the most abundant protein on Earth. However, it's not that efficient and it has an Achilles heel. So in addition to fixing carbon dioxide into RUBP, Rubisco can also bind to oxygen. This process is called photorespiration, and this is bad and it can cause two problems. The first is that the efficiency of fixing CO2 is going to be reduced because some enzymes are accidentally binding to oxygen. The second is that the products formed when O2 is combined with RUBP are not useful in the production of glucose. So to combat this, plants have to shuttle the useless photorespiration products to organelles called peroxisomes. It's here that considerable effort is made to get these molecules back to something that can be used to make glucose. Now, fixed carbon is lost as carbon dioxide in this process, and there is a net loss of carbon atoms. So you may be tested on the different types of photosynthesis. So you have C2, C3, C4, and CAM. You might run into all of these. So C2 photosynthesis refers to photorespiration because a product of this unneeded process has a molecule with two carbons. So C2 photosynthesis, just think photorespiration. Now C3 photosynthesis is normal photosynthesis where 3-carbon PGA is produced and used to make glucose. The majority of plants are C3 plants, which have no evolutionary characteristics to combat photorespiration. And then there's C4 photosynthesis, and the whole point of C4 photosynthesis is to prevent photorespiration. To do this, the light-dependent reactions and the Calvin cycle are physically separated from each other and take place in different cells. So we first start in a mesophyll cell. Now simply put, these are the cells that undergo photosynthesis. Now it's here in the mesophyll cell that an enzyme called PEP carboxylase will fix CO2 into a 4-carbon molecule that we've probably seen before, and that's oxaloacetate. We've seen this in the citric acid cycle. Oxaloacetate can be abbreviated OAA. So OAA is then converted into malate, and malate is then moved to a specialized cell called a bundle sheath cell. So it's shuttled out of this cell into another one. So here, malate is broken down into pyruvate and CO2. All you need to know about this pyruvate is it's going to go back into the first mesophyll cell again and be converted into PEP by way of ATP. So the CO2 here in the bundle sheath cell will combine with RUBP via Rubisco and then proceed to undergo C3 photosynthesis. So what is the whole purpose of this process? Remember, it's to prevent photorespiration, and photorespiration is when oxygen attaches to Rubisco. So bundle sheath cells are surrounded by mesophyll cells, and thus hardly any oxygen will be able to get into the bundle sheath cells. So it's here in the bundle sheath cells that Rubisco is active, and if oxygen's not present, it's not going to bind to it. So this is a way that a plant can spatially isolate its photosynthetic processes to prevent photorespiration. Now we get to CAM photosynthesis. This is another way that some plants prevent photorespiration. This process is near identical to C4 photosynthesis with a few key differences. So instead of separating the light-dependent reactions from the Calvin cycle spatially, CAM plants separate these processes in time. So this is called temporal isolation. So stomata are these pores found on the bottom of leaves. These are little holes and are the site of gas exchange. This is how CO2 gets in and O2 gets out of the plant. If you ever run into guard cells on your exam, just know that these are specialized cells that surround each stomata and they control whether it's open or closed. So during the day of a CAM plant, the stomata are going to be closed to prevent water loss. Now the process of water evaporating from a plant is called transpiration. So the stomata are closed to prevent this. However, during the night, the stomata are going to be open to let in CO2. PEP carboxylase then fixes CO2 into PEP, just like C4 photosynthesis, to produce OAA and then malate. Now, unlike C4 photosynthesis, in CAM plants, the malate is stored in the vacuole instead of being shuttled off to bundle sheath cells. Now, as the night passes and it's daytime, during the day, the stomata are closed, and the stored malate is going to be converted back into oxaloacetate, OAA, and that is going to release CO2 and PEP. So now the plant has CO2 for use in the Calvin cycle. And that is how CAM plants temporarily prevent photorespiration. Now I know that's a lot of information, so if you're crunched on time, just remember this quick overview. C2 refers to photorespiration. C3 is normal photosynthesis. C4 mitigates photorespiration by separating the initial CO2 fixation from the Calvin cycle. And it's dealing with bundle sheath cells. So C4, think bundle sheath cells. Then lastly, CAM photosynthesis is dealing with the day and night cycle where the stomata are going to be open or closed. So think of a camera, right? And photographers are always worried about the light during the day and the night to get the perfect shot. So that's it. That's photosynthesis. Good luck.